Hi, this is Amy Proal with the Polybio podcast, and my guest today is Tobias Lanz. And he is an MD and academic research scientist in the laboratory of Professor William Robinson at the Department of Rheumatology and Immunology at Stanford School of Medicine. And Dr. Lanz's research focuses on B cell biology in autoimmune and infectious diseases, including multiple sclerosis or MS, lupus, rheumatoid arthritis, and COVID-19. And explores viral and microbial triggers of autoimmunity. And recently, Dr. Lanz was the first author of a breakthrough study that connects Epstein Barr virus directly to the MS or multiple sclerosis disease process, a finding that we'll discuss in depth in today's podcast. And with that, Tobias, thank you so much for joining.
Thank you, Amy. Thanks so much for having me. Happy to talk about our study.
Awesome. It's an incredible study. When I read the paper, it really was one of the studies that I think has most provided clarity on a concept called molecular mimicry. And I'm just going to try to explain that quickly in simple terms, where researchers who are studying and trying to connect the activity of pathogens to autoimmune disease processes often recognize that in patients with autoimmune disease, there are antibodies that target self, right? But the million-dollar question is, where did those antibodies come from? And why are they generated? And one of the ways that they can be is because the immune system targets a protein created by a pathogen. And that protein has a similar size and shape to a human protein or receptor structure. And so the antibody then cross-reacts and targets that human protein, sort of as collateral damage. So that's what you demonstrated in this study with Epstein Barr virus and MS. And with that, let me have you better explain what you found.
Well, I think you touched on it very well and explained it very well. There are two main problems with research on autoimmune diseases as I see it, or with the aspect of antigens in autoimmune diseases. And one is that for many autoimmune diseases, we don't really know what the autoantigens are. There are a couple where it's very specific. For example, myasthenia gravis, there's the acetylcholine receptor is really targeted, or MuSK is really targeted, and it can take out the antibodies from patients, put them in a mouse, and they get a similar mouse model of a disease. Or pemphigus is another example for a very specific autoimmune diseases. For many other autoimmune diseases, including multiple sclerosis, it's not as simple, and people have been researching for decades what exactly are the antibodies, and let's not forget the T cells. What exactly are the T cell antigens that really drive these diseases? And I think the overall consensus is that it's probably not just one antigen. There might be a triggering antigen at the beginning, but then there's epitope spreading, which is a phenomenon that if you have a reactivity against one molecule, then over time the reactivity, or the spread of the broadness of the reactivity spreads, and you have more reactivity against multiple antigens. And that probably happens in many of these autoimmune diseases. And we end up with taking snapshots of patients, trying to study what exactly is the reactivity against, and then having difficulties because we find in some cases hundreds of glial antigens that seem to be more prominent in MS patients than in healthy individuals. And then the other aspect that you mentioned adds to that. What is the trigger, the initial trigger of this antigen or antibody reactivity, T cell reactivity? And one major mechanism that we showed in our paper is that pathogens, viral pathogens, but also bacteria and other pathogens could really trigger an immune reaction. And as you explained, we have stimulation of B cells, T cells, innate immune cells that react to the pathogen, exactly what they should do. But then those activated B cells and T cells also react to another a self antigen, and basically there is a breakdown of the tolerance mechanisms that usually prevent us from developing immunity against ourselves, and then this whole system results in an autoimmune disease.
Yes, No, that was a great way of describing that. And in this particular study, you did some additional work and quite a bit of additional work to really clarify the relationship between a specific Epstein Barr protein and a human glial protein. Can you describe that specific relationship further and how you were able to characterize it?
Sure. Well, first we kind of set out with taking a different approach to what people have done in the past. And the traditional approach to how to find antigens and to how to identify molecular mimicry is basically taking serum from patients or taking spinal fluid, CSF, from patient, and stain histology slides with it, or throw them on large protein arrays where there are like hundreds of different spots of proteins, and then see which antibodies stick to those proteins and which antibodies do we not find in human samples and where they don't stick. And that is a quite promising approach and had some success, but it is also quite diluted because as you know, in serum and spinal fluids, there are thousands or millions of different antibodies targeting different things. So it's it's a very noisy system. So what we did in our study is, instead of taking the spinal fluid or the serum of the patients, so instead of focusing on the secreted antibodies, we focused on the B cells, and the B cells are the immune cells in us that produce the antibodies. And we used a methodology called repertoire sequencing. Maybe we should explain it. So a B cell produces the antibody, and our body has the capacity of producing 10 to the 14 different varieties of antibodies. Each B cell produces only one kind of antibody, and this large broadness of reactivity of antibodies is necessary so that our bodies can basically defend against any pathogen that comes our way. However, we only have something like 20,000 genes. So how does our DNA encode for such a large variety of antibodies? And the mechanism that applies there is that B cells mutate their DNA. Usually all the DNA in our body is considered to be the same in every cell, but in B cells and in T cells, there are a couple of enzymes that mutate the DNA that encode for those B cells and B cell receptors. B cell receptors and antibodies are the same gene. And those B cell receptor genes are segmented. So there are segments called V segments, D segments, and J segments. And during B cell development, those segments are shuffled together. And then you can imagine if you have something like 70 V genes, I'm not sure, 50 D genes, then you already have, and you shuffle them all together, you'll already have a variety of a couple of thousand different antibodies that are possible. And this mechanism happens in two chains, the heavy chain and the light chain. And then on these junctions and in the other regions too, but mainly at the junctions between those segments, there the B cell introduces additional mutations into those B cell receptors and antibody sequences. And then a third mechanism happens once the B cell has developed, and then the B cell recognizes an antigen. So let's say a virus comes in, the B cell is selected and says, oh, I'm the B cell that really can fight this virus. Then the B cell proliferates and gets activated, and then this machinery of mutations turns on again, and in an effort of the B cell to really develop even a more specific antibody against this pathogen. And so the whole entirety of all the B cell receptors and antibodies that our body has is called the B cell repertoire, and in conjunction there's also a T cell repertoire. And we set out to sequence the B cell repertoire in MS patients, and we did that from the B cells that we found in the blood of these patients, and we did that as well from the B cells that we found in the spinal fluid of the patient. And the spinal fluid is important because it surrounds the brain and it surrounds the spinal cord. So it is basically in direct proximity to the central nervous system where the pathology happens, where the immune system fights those or creates lesions that lead to the symptoms of multiple sclerosis. And usually there are hardly any cells in the spinal fluid. In MS patients, there are a couple of lymphocytes, not many, that made it a bit tricky to fish out and then single cell sort the single B cells from the spinal fluid. And then, so our methodology was we single cell sorted the single B cells and then sequenced each single B or the B cell receptor, the antibody of each single B cell. And it is necessary to sort these in single B cells because we need these two chains of each B cell, right? So if we were to sequence like 1,000 or 1,000,000 B cells, we can basically only say, okay, these B cells have these heavy chains and some B cells have these light chains, but we can never match the light chain to the heavy chain, and we can never have the complete information of each antibody molecule. And when I started in Bill Robinson's lab here at Stanford, where the study has been performed, Bill's lab had a quite unique methodology to do this, to do the single cell sorting and then sequencing of these B cells. Now technology has a bit evolved, and it has become a bit easier to use droplet-based sequencing methods to sequence the repertoire of B cells. But that was a long story, but what that allowed us to do was to not take the spinal fluid with 1,000 antibodies or millions of antibodies, and but to focus on single antibodies in this repertoire. And we chose B cells that were clonally expanded in the repertoire. So that kind of gave us the indication this particular B cell has expanded. So there we don't find one B cell, but we find 10 B cells or 20 B cells with a very similar sequence, slightly mutated. And we predicted that this B cell stems from a progenitor B cell, which likely encountered some self-antigen and started proliferating and mutating a bit. So we produced as monoclonal antibodies about 150 of those B cell sequences, and then tested them with all the methods that we had at our disposal. And it took me a really long time so the sequencing part and the sample collection part, sorting, sequencing was fairly straightforward. But then I thought it would be way easier to find target antigens for these 150 antibodies. And that was quite the tricky part. So we did immunoprecipitations, we did 2D gels, we did a phage display in cooperation with UCSF here, which actually is also in the paper. And then we did large planar microarrays, and those microarrays are basically histology slides where you spot hundreds or even thousands of different proteins on them, and you put your monoclonal antibody on those slides, and then you try to find out to which spot the antibody sticks. And we became more and more interested in antiviral immune responses. And so we produced a virus microarray where we had protein spots from and viral lysates from multiple viruses that are implicated in the pathology of multiple sclerosis. And those are mainly EBV. So we had about 300 peptides from the EBV virus. EBV is the Epstein Barr virus, and about, I'm not sure, 25 proteins of the virus and viral lysates. But we also included CMV and other herpes viruses and mumps, measles, rubella, and human endogenous retrovirus, or at least one human endogenous retrovirus. And the very interesting thing that we saw is that we saw that a couple of our antibodies really bound tightly to EBV antigens. And we focused then for the rest of the paper on one antibody that really focused, that really bound to one specific antigen in EBV, and that is the EBNA1 transcription factor. Yeah, okay. You want me to continue for the?
No, that's, I mean, you know, there's no way, it's technically complicated what you did, but there's no way to simplify it. And what I think that some of the takeaways from what you said are, is, first of all, it's really amazing to hear what is capable of being done in a lab right now. So just, you know, a couple of years ago, the single cell sequencing and the ability to understand what's happening in a single cell was rarely, you know, it's really used, and you are started to be able to really, you know, work in that capacity to be able to use some of these very, very targeted tools in a really clear fashion through a very, like, delineated series of experiments. I think that one of the things that stands out is you were, you know, able to, I guess let's focus on the way that you were able then to isolate. Overall, you were able to find that one of the Epstein Barr proteins, the EBNA1 protein, was a mimic for a glial protein, right? And you even came up with the crystal structure of that protein to show that, right?
Yeah.
So how did you do that? Yeah.
To summarize that, so what the advantage of our approach was, what my long speech from just a minute ago was that we could, instead of just having a broth of thousands of specificities, we could really select for the important ones and then find the antigens to those. So yeah, exactly. So then once you have one antibody and can study just one antibody, it becomes way easier than if you always have to work with 1,000 or more specificities. So as you said, we identified then reactivity against this special EBNA1 transcription factor. And what we also could see is that we did not just find specificity of this antibody to the whole or to anywhere in this transcription factor, but specificity was directed against a certain very short region in the EBNA1 transcription factor that has been described in about five papers before, that MS patients have a higher reactivity against this particular region than healthy individuals. And that was an indicator that it should be a highly interesting region. And actually, the new paper that just came out in Science, which I'm sure we'll talk about in a minute too. If you look in the supplementary tables there, the first hit that they found as being highly reactive in their MS patients is exactly encompasses the region that we also described. So there's something up with this particular region. And then, as you said, of course, we were interested. Did we just now find an antibody that is randomly in the spinal fluid, and that is just a bystander antibody, or has no particular meaning, or do we really found an antibody that detects EBV but also detects the self antigen? And so what we did then, we took this monoclonal antibody and tested it on a so-called uProtarray. uProtarray is a protein array that has 16,000 different spots. I think they updated it now to about 20,000 different spots with different proteins spanning at least a large portion of the human proteome. And there we could pretty clearly see that the antibodies binds to a particular protein called GlialCAM. And GlialCAM is expressed in glial cells, in oligodendrocytes, and in astrocytes. And it is a cellular adhesion molecule, which is involved in the attachment of glial cells to each other, the development of glial cells. And it is also a subunit of chloride channel, and it plays a role in the chloride and water homeostasis of glial cells. There's a very rare neurodegenerative disease called MLC, which is one of the causes is that GlialCAM is mutated, and then you have a disruption of the water chloride, the glial water chloride homeostasis, and then you get edemas in the brain neurodegeneration. So yeah, and then we continue. So we saw that GlialCAM has a region that closely resembles the region of EBNA1, but it's not completely the same. And what we did then is to better understand which amino acids are really important for the binding of our antibody to that antigen, and how is it that this slightly different protein slightly different protein or slightly different peptide binds to EBNA1 and to GlialCAM. We made a crystal structure of the EBNA1 peptide in complex with our antibody. And we on the crystal structure, you can easily and very nicely see which hydrogen bonds build out between the amino acids of the heavy chain and the light chain and the antigen.
That's cool.
And what an interesting little twist that we could also find is that the antibody actually does not bind to GlialCAM just as it is, but GlialCAM needs a phosphorylation at a certain serine residue that enables this binding between the antibody and GlialCAM. And I think that, or I'm planning to follow up this in more detail, because I think that would be a really nice mechanism that could add to this molecular mimicry, because phosphorylation or other post-translational modifications are modifications on the protein. So the protein is translated, and then afterwards phosphates or different groups can be added and cut off again. And that can be very organ specific. So it could be very different in the brain than it is in the lymphoid organs where the B cells mature. So it could be that those post-translational modifications are a reason for breakdown of tolerance for B cells and T cells, and that could add to the mechanism.
Yes, that was a really cool part of the paper. I agree, where, I hadn't even thought that far when I was thinking about molecular mimicry, just as a concept more, when I'm thinking about that process. I hadn't really thought about the fact that the post-translational modifications might also be part of what eventually led to the, real targeting of that protein, that was part of it. So, you really, in this study, were, I think, one of the first teams to really flesh out the process of, the sort of antibody being identified in the first place that targets the EBNA. I usually call it EBNA1, but you're calling it EBNA, which I will now call it now it's EBNA. I'm EBNA1, EBNA, and then
it's shorter, yeah.
Right, and then able to show that that there was even modification in the way that protein was finally afterwards post-translational modification. In other words, you went far beyond just the simplistic sort of, hey, this might have a similar size and shape to this. You really fleshed out the intricacies of each part of the process and how that can occur in a way that I think makes it so real to a person who really digs through the paper, where all the little bits of each of the process are able to be better understood. So you really added to.
That was mainly possible through this crystal structure, because there you can really see what interacts, because you can blast basically lots of sequences and can try to predict which antigen might be prone to deliver molecular mimicry. But as we can see in our study, and everybody who opens up the figures, there's one alanine in the peptide region that we defined, and the alanine is there in the GlialCAM, and the arginine is there in EBNA1, and otherwise the peptides look pretty similar. But if you look at the crystal structure, you see that the arginine, the side chain of the arginine reaches deeply into the binding pocket and has two hydrogen bonds with the heavy chain, versus the alanine doesn't have much of a light chain that could mimic that. So then we could see that the post-translation modifications is actually 5 amino acids upstream and terminal of the disrupted alanine. And we don't really have a crystal structure of GlialCAM yet, but we believe that this polar phosphate side group would enable interaction with the heavy chain again. And we also tested, so with the repertoire sequencing, what I just explained, you can also see the evolution of those antibodies. So basically, a naive antibody that has none of these mutations that I described, that naive antibody already binds to EBNA1 with pretty much the same affinity as our mutated antibody does. However, it does hardly bind to GlialCAM. So it needs those mutations, so those additional mutations and this additional development to generate the reactivity against GlialCAM. So what we believe is, at the beginning, there was a naive B cell that detected EBNA1 or detected the viral protein, and then got activated, started to mutate, somehow made its way to the brain. We unfortunately don't know how that, what facilitated that, but probably then in the brain, it encountered GlialCAM, started to mutate further, got selected for specificity to GlialCAM, and then had at least some impact on neuroinflammation. And we could show that impact of neuroinflammation as well. So we took EBNA1, the peptide that we identified, injected that into mice, immunized mice with it, and then on top of that, induced a common mouse model of multiple sclerosis, which is called EAE. And we saw that the mice that were injected with EBNA1 had a more severe disease course in the EAE model than the mice that were not, or that were injected with the control peptide.
Yeah, that was a really cool little follow-up experiment to kind of solidify that. Yeah, with the crystal structure, that's, you have a 3D structure then, to be clear. That really allowed you to better understand what was going on. Are you planning on making more crystal structures for other viral proteins? It seems like there's so much opportunity there.
Yeah, that's true. Well, I'm not sure if for my current project doesn't include viral proteins in particular, but I'm pretty interested how GlialCAM itself looks like. Yeah. So I'd like to follow up on GlialCAM and include several crystal structures in a next study that could come out of our lab. Yeah, that's great. I also find that fascinating, and I find it fascinating that I have, that's basically the first crystal structure that I produced, and it was quite nice to cooperate with the MSKC here at Stanford, which is the macromolecular structure knowledge center, who have expertise and you can basically or all the setup to produce your own protein crystals, and then in collaboration with SLAC, the linear accelerator, the physics department here at Stanford, we could shoot those crystals and get the diffractions. And lots of, I need to say this, lots of people have helped me, including the Garcia lab, Kevin Jude here in Chris Garcia's lab, and Daniel Fernandez here in the MSKC were major drivers that helped me achieve this crystal structure. Wow. But I like being able, and I think that's a nice thing about our study, is being able to take patient samples, to have a wide range of methods to take patient samples, sequence the single cells out of these patient samples, get to the very molecular structure and study it on a very molecular level, and then expand it out again and test it in mouse models. And of course, the optimal final result is if we could transform that knowledge back into humans and develop a therapy out of that.
Yes, exactly. That kind of series of events you just described has so much potential for, other targets, other conditions, other diseases that are also connected to pathogen activity, autoimmune conditions, but that have infectious triggers that we better understand, to the point where, I feel like when I think of the sort of clonal expansion that you study or see in that paper, I'm like, we need to clonally expand you, like clone you, so that you can just start to create crystal structures for all the
nice.
So clonal expansion of that exact series of experiments but for an even wider range of different pathogen proteins and, targets and like that. That just seems like the future, to be honest. So it's really cool. So I guess, some overall takeaways from this then is that, a lot of times we're looking in autoimmune disease for root cause, right? And so in autoimmune disease, then in this particular study, it seems that the Epstein Barr virus and it's, the proteins it's creating are the root cause of the disease. Would that be accurate to say? Because it seems like if you were able to get rid of the virus, then none of the rest of the, process that you described would occur, right?
That's correct for the process I described so far. Yeah. I think we should touch on the other paper that came out almost at the same time as our paper, because I think that paper makes your point pretty clear, right? So you want me to briefly describe it? Yeah. So that paper came out like a week earlier than our paper in Science, and it's a huge study of about 10 million patient, or let's say 10 million veterans. And apparently in the American military, you give blood at multiple time points, and those blood samples were stored. And this group in Harvard was able to get access to some of those patient samples. And they studied, they identified all the patients that developed MS out of these 10 million veterans during their time in the military service, and they identified, I believe, 955 patients, and they got, I think, 801 sample materials from 801 patients, and then they checked how many patients were EBV positive versus control samples in the same cohort that did not develop MS. And so they found 35 MS patients that were actually not EBV positive. But then they had, that is super nice of the study, they had previous samples and could see, okay, when they entered the military service, they were EBV negative, but did those patients actually get infected with EBV before the onset of their disease? And all but one patients are actually positive for EBV before the onset of the disease. And they, of course, did the proper controls. They checked, is that just with any virus, like this? And they checked CMV as a comparative virus, and that's not the case. They checked in their control group. Just anyone in the military get infected with EBV. That is also not the case. So it seems really clear that EBV, that basically, I think one can say 100% of MS patients are infected with EBV. And previous studies have shown that too. It's just a very hard problem to prove. And the main reason for that is that 95% of all of us are also infected with EBV, and we luckily don't get MS. So it's hard to prove that if you have only a comparator of 95 to 100, it's really hard to prove that this is a true effect. However, and other groups have shown similar things in the past. There was a paper like about 2 years ago from Ruprecht, and I think he's in Berlin, that also showed that 100% in I think a cohort of 900-ish patients were positive for EBV. MS patients were positive for EBV, and they checked also for EBNA1 reactivity, and then a couple of patients were negative there, and then they checked for other antigens and could show that they that actually everybody was positive. But it is hard to really show a difference in only 900 patients, and then of course 95% of the 900 controlled patients is also positive. So that is, I think now with this new Bjornevik study in Science, it's on really solid evidence that EBV has something to do. And I think the way we should think about EBV is now that it's a prerequisite for developing MS, but it's for sure not the only trigger because otherwise all the healthy individuals should also develop MS.
Right. That is the first, because exactly, it was cool to see that Harvard study come out first and kind of show that really clear association between MS and EBV just directly in such a large, within such a large number of patients, as you mentioned in that study. And then your study come out the next week and kind of come out with this mechanism, this direct mechanism by which, the immune system targeting an Epstein Barr protein can lead to then a cross-reactive targeting of human protein or glial protein, right? Yeah, they were it was like, wow, the first Harvard study was, good enough. But then the second, yours bringing in that mechanism on top of it was huge. So all of a sudden there was really this, big push to say, wow, look at the contributions of Epstein-Barr virus to this disease. But that being said, when I start to just talk to people about the findings, that is sort of the first question that comes up is, well, a lot of people have EBV. So why doesn't everyone get MS? And I suppose then one of the reasons would be perhaps the immune system is not in the mechanism you described, this mimicry, maybe that's not happening in everyone, or perhaps the virus has to be in a certain body site or, active in a certain way for that process to begin, or perhaps there's a human genetic component, human genome predisposition that facilitates, also has an effect on the immune system in a way that allows, the EBV phenomenon to move forward. I don't know. What are your thoughts on that? Why doesn't everyone with EBV get MS?
I think all of the above what you said is probably true. Our mechanism, we could show it for about 20 to 25% of the few patients that we checked out. And we have 3 different cohorts in our paper. One is like 35 patients and 2 are about 70 patients large. So not the largest cohorts, but we did find a significant difference between MS patients and the control patients that we had. But what you said is absolutely correct. So on top of having EBV, and then maybe to add to EBVs, a couple of additional points of evidence point to EBV. So if you have symptomatic EBV infections, which is infectious mononucleosis, mono kissing disease, then you have an even 2.5-ish higher risk to develop MS. But on the other hand, there's also a high risk genetic risk factor for MS, which is HLA-DR15, which is the most prominent risk factor. And then there have been large GWAS studies been done on also huge MS patient cohorts, which show about 230 risk genes which are associated with us. So there's definitely a genetic risk factor. Also monozygous twins, about a quarter of monozygous twins get MS. So if one has MS, you have a 25% chance that the other one gets MS too. So you could say that maybe 25% of your risk is genetically programmed. That's a very rough estimate, yeah. And then other environmental risk factors are also known. The one very prominent one is vitamin D, that is always discussed. So there's a very strong imbalance in distribution in the world of MS. So the northern hemisphere and the southern hemisphere has a lot of MS incidence and prevalence, versus the equator region in the world has little MS incidence, and that is often attributed to sunlight and vitamin D exposure. Then there are also epidemiological studies which show that if you are in a high-risk country before puberty and then move to a low-risk country, you basically acquire the risk of the new, or vice versa, you acquire the risk of the new country, but somehow after puberty, you don't take the risk of the new country on you. It seems to be a set time frame where you acquire your life's risk of developing MS, and then after a certain age, you move and you don't change your risk. Does that make sense? Was that understandable?
It does. Yeah, it makes sense.
So that could be also maybe it's you have to have enough vitamin D in your youth, or it could also speak for some other pathogenic agent, some virus that only is present in the very high and low areas, and not in the equator region. Maybe a virus that doesn't like warm weather. There might be additional factors that we don't know yet.
Right. Like another pathogen that, for example, might impact the immune response in a way that allows EBV to become more active in these patients, or something like that. Or I guess I also wonder about sort of just the transmission of EBV in certain countries versus others, or just the prevalence of that virus. You know, I know this is bad, but one of the things that I've always, because there's, as you said, there's been some evidence for EBV and MS for a while now. So when I was in college, even, it always made me nervous how little people cared about getting mono. You know, we have a college setting and people are like, man, a bunch of us got mono. And I think, do you think it's time that we have to change our attitudes on that, be a little bit more serious, actually sort of educate students and say, look, getting mono might not be might be kind of a big deal. We have to be a little bit more careful. Because I wonder if there's, transmission where you could have a lot of clusters when you have groups of people trading saliva in that kind of atmosphere. I guess that's also a cultural thing as well, about how much people are even trading the virus. Although I guess we'd have to look into all that.
Well, that's true, but on the other hand, we also want to have fun in college, right? So it's not.
I know. That's really the thing is I agree. There's enough. We already have COVID, SARS-CoV-2, and then we're going to make everyone feel bad about EBV. But I don't know. Yeah.
No, there are studies that say that about 30% of college students who start college are EBV negative, and those are actually also the good groups that are enrolled now in EBV vaccine studies. And then within the first 2 years of college, those numbers dramatically rise. And then, of course, colleges and also apparently the military are pretty concerned, because mono, we consider it not to be the most severe disease, and it's probably not, but there's a chronic fatigue symptom complex around mono, and it is one of the major causes for college students to miss classes if you just start college, get mono, and then really can't perform as well as you were used to.
Yes.
So and the same for the military. I think it's the most common cause to miss months in the military service right after start of the military service.
Yes, the illness that I study most is myalgic encephalomyelitis, or chronic fatigue syndrome, ME/CFS, and that is a condition in which a good number of the cases are initiated by an Epstein Barr infection. So same trend, and exactly, there's probably different genetics and other factors that play into why those patients develop other sets of symptoms than MS. But same trend in which patients can become ill for life after an EBV infection. So that's the first diagnosis that put EBV on my radar, which is actually part of why I'm so interested when researchers connect EBV and explore what EBV is capable of doing in related diagnoses, because obviously the more we learn about what it's doing in MS, the more we can better understand what it might be doing in a diagnosis like ME/CFS, right?
Yeah,
so
that's great. But that also means that, and EBV is connected to so many diagnoses, right? It's connected in lupus as well, as you said, chronic fatigue syndrome, our rheumatoid arthritis, Sjogren's syndrome, and then of course all the EBV related cancers, which I think is a different story. But it also shows that our mechanism and I believe this mechanism is very relevant in MS, as we showed, but it's for sure not the only mechanism out there. There must be either other molecular mimicries or other mechanisms where EBV transforms B cells or other cells to generate different molecular mechanisms that cause all these other associated diseases.
Agreed, because that's the next thing people bring up when I mentioned the connection between Epstein Barr virus and MS is, well, what about lupus? And it's like, no, it could still matter there, right? So there's all these caveats for interpreting this work where I'm not sure. I think it's a University of Cincinnati Children's Hospital study that showed, I think it was EBNA1, could bind directly into the human genome at risk loci for lupus risk and sort of just increase the human, human risk loci, sort of change the gene expression there. Not sure if you've seen that paper. No, I have to look back. It was just basically the overall takeaway was that the viral proteins might be able to alter the risk, the human genetic risk of the disease, change the expression of the human genes.
I need to read up on that lupus paper. There is a study in MS as well, which shows that the transcription factor EBNA2. So we studied EBNA1. There's another.
Oh yeah.
EBNA2 that binds to a myriad of risk loci for MS. About half of.
That's what I'm thinking of. Okay, sorry, that one.
Yeah, about half of the risk loci are connected to this transcription factor and can be modulated by EBNA2, and that is, I think that's a very interesting hypothesis, and also we have to say EBNA is EBV is a huge virus, right? It has over 90 proteins that it expresses in its lytic stage. It is highly tropic to epithelial cells during transmission in the saliva glands, but then in a normal latent stage, it's highly tropic to B cells. So B cells are the main cells that harbor EBV, and also, of course, B cells are the main driver cells of several of these autoimmune diseases, and especially for lupus, there's a large portion of lupus pathology is B cell biology.
Yep.
And EBV has a couple of very, in addition to those transcription factors, a couple of very well-studied molecules that alter B cell biology. For example, LMP1 and LMP2 are two latent proteins that mimic the activation pathways of B cells. So the CD40 activation pathway, the B cell receptor activation pathway, which could be just you have a viral protein on the surface of your B cell that constantly activates this B cell. And that way the B cell does not need the proper activation channels via an antigen that activates the B cell receptor. It gets some T cell help, and then the B cell gets activated and produces antibodies, but it can be basically activated on itself by the virus always pushing for activation. So several of those molecules have been described and pretty well studied. So those could be very or could be very important mechanisms for autoimmunity that make a lot of sense.
Absolutely, yeah.
It is hard. Really, there are a couple of technical difficulties that make it hard to study Epstein Barr virus. One is that the genome and the transcription of the genes is quite low, so you have a hard time detecting EBV genes, especially also in single cells. And another factor is that the EBV infected cells in healthy individuals, but also in autoimmune patients are pretty low. Some studies say like 1 in 100,000 B cells or 1 in a million B cells even could be EBV infected, and it could still be relevant, right? And we have a hard time sorting those EBV infecting B cells and then comparing what does it do in a lupus patient and what does it do in a healthy patient.
Yes, we have a similar challenge where one of the projects we're working on with our organization is to just find the genetic material of EBV and antigen in tissue samples. And the thing is, a little bit of virus can be present in a person, and you may not be in the particular tissue sample you analyze. It's the same thing where you have to collect a lot of tissue to be able to actually find the piece that might have the virus, the genetic material. And in that case, it becomes a pretty big endeavor to be able to get all those samples to be able to do that. And especially for neurological diseases, it's very hard to. Exactly.
to get
But what it shows is that a little bit of virus can do a lot. In other words, I think sometimes when I've talked to people about pathogen activity in chronic disease, they think that almost every cell of a person has to be infected for that pathogen to be driving a disease, but it doesn't seem to be the case. You could have sort of a lower level of virus, but it's doing some very brutal things. Some of the mechanisms we described, where it's more of its activity, like less virus, but evolved to be a big driver of some very clever disease mechanisms in order to do that. Yeah.
And it doesn't even need to be in the tissue
right.
itself that you study, right? It could be in the lymph nodes, where a couple of very rare infected B cells stimulate T cells. That we should mention that as well. So the B cells don't only secrete antibodies, what we showed, but they also are very potent stimulators of T cells. And MS was or is still considered a very much T cell driven disease. So the B cell encounters its antigen with the B cell receptor. Then the B cell receptor gets activated and encapsulates that antigen and eats it, and then presents it on its cell surface. And then the T cell comes from the other side and gets activated that way. So that could be a major mechanism how T cells are activated through EBV infected B cells. Yeah.
That makes sense. Yes, definitely. Well, I think then if you have a little bit more time, just quickly, I just want to say, what does this mean for treatment of MS? I mean, I know that I don't want to get too ahead of everything, but obviously patients with MS thus far are largely given immunosuppressive drugs, which makes sense because you're trying to control the immune system. But now we have this opportunity to possibly target the virus that's basically at the root of a lot of the immunopathology.
Yeah,
so,
no, I totally agree. So that's the main question, right? How can we leverage all this knowledge to really treat MS patients? We should say there are two treatments already, which might by proxy actually have something to do with EBV treatment. So one is a newer treatment, anti-CD20 antibodies, which are rituximab or ocrelizumab, which are very effective in MS patients. And those treatments, it was actually surprising that those treatments work, because as I just said, that MS was always considered a T cell mediated disease. Right. But if you deplete B cells, those are highly effective treatments for MS. And again, probably not only through decreasing autoantibodies, but also through decreasing B cells that stimulate T cells and thereby drive the pathology. Yeah. And it could be, and CD20 is on memory B cells that harbor EBV. So it could be that actually by depleting those B cells, you also, to a certain degree, deplete EBV or EBV positive B cells.
Right, however, you take away its home in a sense. It's in the B cell and it no longer has as many B cells to infect. Or you would take away its home in simple terms, like it doesn't have as many B cells that it could infect and reside in.
It could be, but I'm not sure if you can deplete the whole virus. If you're definitely not getting rid of the whole virus.
Okay.
Is that your question? No.
Well, it would, in other words, it would just, if your Epstein Barr virus tends to hang out in the B cells as well, to persist in them, right? So in other words, it would have less cells in which it could persist.
Yeah, exactly.
Okay.
Or
okay, right.
if we're sticking to that paradigm that we discussed a minute ago, if your EBV positive B cells are the drivers of the disease, and there might be, as we said, just 1 in a million, but you just deplete all the B cells, you might just get rid of the one B cell that is EBV positive and does something. And another very old medication for MS is interferon beta, and interferons are antiviral cytokines, and was actually at the very beginning, I believe, invented by thinking that MS could have something to do with viruses and infect the brain, and then interferon gamma and interferon beta were tried. Interferon gamma makes disease worse, but interferon beta seems to be a quite potent quite potent treatment for MS. So I'm not. It's unclear how much interferon beta treatment really interferes with EBV, but it's another indication. And then in terms of what treatments are ahead of us, I think one very obvious one would be a vaccination against EBV. And now in recent months, that idea became even more traction. Moderna just started a clinical trial for a vaccine against EBV using their mRNA technology that is now proven with COVID-19. And that seems to be a very exciting prospect. But also there have been a couple of studies already going on for the last decades, which try to vaccinate against EBV. And the way Moderna is doing it, they encode in their mRNA the 4 glycoproteins, which are at the outside of the envelope. So they really want to target the virus when it comes in, and it'll be met with antibodies and hopefully eradicated immediately. Other studies, and that might also be approaches that might be valid, actually then target proteins that are expressed by the virus in the cell. And you could imagine that those would be treatments that are even valid in a patient that has MS already. Yeah. Does that make sense? Because you're not only targeting active virus particles, but you would basically target an EBV infected B cell. However, and I think that's another study or another point where our study comes in handy. You would, of course, like to avoid those antigens that we have described. So you wouldn't want to immunize with the EBNA1 region that we described, where all this molecular mimicry is happening. And I think our study and other studies that showed molecular mimicry for certain EBV proteins guide the way a bit for these vaccine developments to tell them stay away from those proteins.
Absolutely. I think that's one of the most important things coming from your work is that when I first heard about Moderna's vaccine, I just heard there was a vaccine. And I said, what epitope are they going to use? And I said, please don't be EBNA1. I figured they'd be smarter than that, to be honest. And it's surface proteins, which sounds more promising. But
those surface proteins make a lot of sense for.
Exactly. That's the good. Sounds like a good target. But the capacity to really, really be careful on that front by just doing the kind of experiments that you did in the paper we described today, to just be able to see the capacity for mimicry for a particular epitope that, in any of the mRNA vaccines that are going to be developed as we go on, because I think they're going to develop them for many pathogens, right?
Yeah.
It just seems so important and so clear, just picking the epitope to express in the vaccine that will be least likely to, have issues with mimicry, and then just make the vaccines, as safe and effective as possible. So I think that's a really big other important takeaway from your work is the ability to do more of that moving forward.
It'll be really interesting to see how that turns out and how effective it is. The other question is if it will be possible to achieve real sterile immunity. So a patient who has the vaccine will be really protected and will not get the virus, or if it is like in other vaccines that you will still get the latent virus, but the outbreaks, the lytic transformation of EBV will then always be suppressed. And to me, it's currently unclear if that would be sufficient to really suppress MS. I think we'll see.
Right, just because some of the proteins that MS that you implicate are sometimes expressed during latency, right? So it's hard to say.
EBNA1 is the one one of, let's say, 3 proteins that is really expressed during latency.
I know. These viruses, they're so tricky.
But other approaches might be very valid as well, right? So you could think about antivirals and anti-herpes antivirals have been already even tried in smaller MS studies with limited success. You could think of if you know the antibodies that we described, you could think of mutated antibodies, basically that inhibit the interaction between those pathogenic antibodies and the target antigens. So there are multiple ways how you could target. You could also induce tolerance against those antigens, against the EBNA1 antigens or the identified molecular mimics. There are multiple ways how to induce tolerance. Even BioNTech, the company that developed the Pfizer vaccine, also tweaked their mRNA molecule to produce a tolerizing vaccine against MOG, which is another myelin protein. And they tried it out in mice at least, and that seems to work splendidly well in those mice.
Wow.
So those might be additional options how to improve the disease with vaccine approaches.
Yeah, those are some promising ideas. And I think, what's great about the work that you did is the more that you understand mechanism, the very specific mechanisms of the disease process, the more you can come up with targeted interventions. So the fact that you're really fleshing out mechanism then will, probably lead to many more teams and people coming in with good ideas for how to mitigate that. So I think that's really cool. Another reason why your work was just really, really great. So I better let you go. I've already asked you so many questions, but no, it was really beautiful work and very cool to see. A lot of us have watched as EBV has been connected to MS, but to better understand just, one of the clear mechanisms as to why is really, really exciting. And it just also offers so much hope that more specific mechanism could be identified in related conditions or with related viruses, and just opens up a lot of possibility on moving forward in a way that can eventually hopefully help patients. So
exactly.
Congrats.
We're also very excited about it. And yeah, we should talk again once about a follow-up study. We hope that
yeah,
we can develop it further.
Absolutely. I would love if you could come back on later and just give some updates. Yeah. Okay.
Sounds
All right.
great.
Well, thank you.
Thank you so much.
All right. Take care.
Bye-bye.
All right. Bye.